Hello everyone, and welcome to this podcast. This is the second podcast on cell differentiation. In this podcast, we're going to focus on how to maintain the pluripotent state, and then how do cells move past the pluripotent state, and ultimately to a terminally differentiated cell. So let's go ahead and get started. Now I'd like to move on to a new topic, and that is the topic of maintaining pluripotent cells. How does the blastocyst, this inner cell mass of the blastocyst, maintain pluripotent cells? And we can also think about how can we artificially maintain these pluripotent cells. To remind you a few things about the blastocysts, they enter this around day 5, and they remain blastocysts until around day 8. These are our embryonic stem cells. I've said that once or twice. And as I said on the last whiteboard, they can become ectoderm, mesoderm, or endoderm cells, which then can go on and form any tissue in the body. They can have continued self-renewal. The reason that these cells can maintain this pluripotent state is their gene expression program allows any gene to be expressed. I forgot to remind you earlier, but on a previous podcast, we had talked about how up until this stage, and during most of this stage, the DNA has been demethylated. And we'll talk in a moment how this DNA will become remethylated during the blastocyst stage and as it exits the blastocyst stage to the gastrula stage. But right now, these cells have a pretty open program to express any gene, with the exception of those genes that have been imprinted. So how do they maintain this state molecularly? So what's the molecular mechanism? And we can break this up into two big sections. And the first is that there are specific transcription factors that are expressed in these cells. And the two main ones are OCT4 and nano-G. In fact, these two transcription factors alone, as hopefully we'll talk about later, can convert any cell in our body into becoming an embryonic cell. Artificially, that is, not naturally. However, that doesn't explain everything, because we can look at one set of embryos and between embryonic stem cells, and we can see slightly different expression profiles. And so the other thing we have to consider, not shocking, since this is an epigenetics class, is that there are epigenetic markings that we have to consider as well. And these epigenetic markings help explain gene regulation in these embryonic stem cells. They help explain the self-renewal of this population of stem cells. They help explain the maintenance of pluripotency, and they help explain lineage-specific differences. And what I mean by this last one, lineage-specific differences, is as these cells differentiate into ectoderm, mesoderm, and endoderm, and become further differentiated into specific cells, epigenetics can explain how certain cells are destined to become specific cells, like the neuron or blood cells. So let's talk a little bit more about the epigenetic programming of these embryonic stem cells. Not surprisingly, as they exist, this chromatin is very dynamic as it goes through these differentiation and as it leaves blastocysts to gastrula. In these embryonic stem cells, H1, H2, H3, and HP, the heterochromatin forming protein 1, are all loosely bound to the chromatin. This suggests that they are in this euchromatin state, meaning that any genes here could be expressed. Doesn't mean they are expressed, because transcription factors need to come along as well, but they have the potential to be expressed. It's sort of like starting the genome with this blank slate, and then as they differentiate, they will take that blank slate and selectively bind H1, H2, H3, and HP1 more tightly. and more permanently as well. That is, they are setting the heterochromatin. As these cells become differentiated, as they move on through gastrulation, genes will selectively be converted into heterochromatin that are not supposed to be expressed. So you would not want genes important in kidney function or kidney formation to be expressed in the ectoderm. So they would be formed into heterochromatin. Due to the epigenetic nature of forming this heterochromatin, they'll stay as heterochromatin for the rest of that cell's lineage. So not surprisingly, what we see here in these cells as they become more differentiated, we see more H3K9 trimethylations, which signifies gene repression, and we see less acetylation of H3, histone H3, and histone H4, which is also a sign of gene repression, and also a sign of less euchromatin and more heterochromatin. And let me just write it here. We see more H3K9 acetylation and more H3K4 methylation, both signs of euchromatin formation. Now I want to introduce this term here called bivalent chromatin domains. So to introduce these bivalent chromatin domains, what I want to do is just make two genes. There's going to be more than two genes involved, but this just simplifies it. This is a gene. This first one here is going to be a pluripotent gene. A gene that is important to be expressed in these pluripotent stem cells. So maybe like OCT4. And this gene here, let's just pretend it's a endoderm gene. A gene that's important to help establish the endoderm and maybe important in establishing cells of the GI tract. So we'll just put a GI gene. Now, not surprisingly then, in the embryonic stem cell, this gene will be expressed and this gene won't. And they'll have the classic epigenetic markers on them to allow this gene to be euchromatin and this one to be turned off. Then as that cell differentiates, the reverse occurs. And we'll call this an endoderm cell now. This pluripotent cell will now not only be turned off, it will be wrapped up tightly into heterochromatin. Whereas this endoderm gene will be turned on. And again, we would see all the classic epigenetic markers on here to permanently turn this gene off and then to actively turn on this endoderm gene. And now every time this cell divides, it will have this gene set. So we have set the lineage of this cell by permanently turning off the pluripotent gene and turning on this endoderm specific gene. This is also a reason why we can say that we don't ever go back in this direction, because we have formed heterochromatin around this gene. Now, the interesting thing about this none of this should be shocking to you as I've just described it, but the interesting thing is that these genes are often found close to each other. And one of the reasons to explain this is it allows the cells to rapidly respond to turn on these endoderm genes or whatever specific gene you might have, even though it is near and closely associated with these pluripotent specific genes. So the bivalent nature, again, allows the cell to respond quickly to turn genes on and off. And I think when they discovered this at first, it was a little surprising that some of these genes were so closely associated with each other. In this part of the podcast, we're going to talk about how we go from a multipotent stem cell to a fully differentiated cell. And if you remember on the last slide, we ended up with this cell at gastrulation, where we had our trophoblast cells surrounding it, and then we had our inner mast cells that had formed three specific tissues. And remember we said this top one was the ectoderm, this middle one was the mesoderm, and this bottom one was the endoderm. And we said these were multipotent stem cells. They had very finite potential to differentiate into specific cells. And if you remember, one of the cells that we said the ectoderm could differentiate into were neural cells. The mesoderm can differentiate into blood cells. And then endoderm would specialize into certain cells like the GI tract and others that we talked about. But I'm going to focus on the top two. I'm going to talk about an example of how neural cells are formed and fully differentiate, and then how blood cells fully differentiate. So as we begin our discussion of neurogenesis, let's remind ourselves that those cells that will give rise to the nervous system come from the ectoderm. And remember, we said the ectoderm contains multipotent stem cells. Now, each of these multipotent stem cells gives rise to a specific set of cells. They can't give rise to all of the differentiated cells of the ectoderm, but each one of these will give rise to a specific pathway. So some of them will give rise to skin cells. Some will give rise to pigmented cells. And others will give rise to neurons. And it is these multipotent stem cells that give rise to neurons that I want to focus on. And those cells are specifically called neural progenitor cells. They're characterized by having a specific marker on them that only these multipotent stem cells have, and that is the marker of CD133. And it's on the surface of this cell. This is a quick and easy way that we can identify multipotent stem cells, and more specifically, we can help identify neural progenitor cells with this marker. Now, how are we going to go from this neural progenitor cell all the way to a fully differentiated nerve cell, which can be easily distinguished from these progenitor cells because they don't have CD133, but they have another marker, and it's called tubulin beta 3 in the inside of the cell. And this is a good marker for a terminally differentiated nerve cell in this pathway. Well, within this neuroprogenitor cell, we will see a downregulation of pluripotent genes. So those genes that help establish a cell as being pluripotent, those have been turned off and have been wrapped up in heterochromatin in a permanent state so that we cannot go backwards, because remember, differentiation is unidirectional. And then we're going to see an upregulation of neural-specific genes. And these include SOX1, SOX3, PAX7, and nestin. As you might predict, these pluripotent genes that have been turned off don't have H3K4 triple methylation. Remember, that's a hallmark for genes being turned on in those regions. Now, these genes will have more H3K4 triple methylation. Now, often in these regions, going back and forth between them, it'll have the two methyl groups. And if you remember from way back when we first introduced this topic, having two methyl groups at this position leaves those genes poised so they can be rapidly turned on. And so it's not uncommon that prior to getting that third methyl group, they maintain this poised state of having two methyl groups there, so they can quickly become expressed. As these transcription factors are upregulated in these neuroprogenitor cells, a process of cellular differentiation occurs, allowing other genes to be turned on and other genes to be permanently placed into that heterochromatin state. And ultimately, you will get into this fully differentiated nerve cell. And again, remember, there's no going back. Once a cell has been differentiated, it doesn't go back under usual circumstances. In our next example, let's talk about hematopoietic stem cells. These are stem cells that will give rise to blood cells. And remember that blood cells arise from the mesoderm. Just like we saw in the ectoderm, it contained many different multipotent stem cells. The mesoderm also contains many multipotent stem cells. One of these stem cells is the hematopoietic stem cell. Now, this can give rise to several different lineages of cells. It can give rise to erythrocytes, our red blood cells. It can also give rise to the lymphoid cells, like our white blood cells. Now, within the hematopoietic stem cells, it's important to remember they can get converted into several different cell lineages. And to help achieve that, several genes specific for each cell lineage are in a poised position, meaning that they are ready to be turned on once they get that final signal. And if you remember, that poised position, what's the hallmark of that, is having that histone 3, lysine 4 double methylated, having 2 methyl groups on it. Because remember, when you add that third methyl group to it, that's when gene expression can occur. So it's in this poised position. And this is a way that it can commit to multiple different pathways in a fairly rapid response. As a specific example, we can look at these 2 genes found in mice, VPRE-B1 and gamma 5. In the embryonic stem cells, prior to forming the mesoderm, these genes are acetylated and have that H3K4 double methylation on it. Remember, in the embryonic stem cells, everything is euchromatin. So this is not surprising. But it's an example of how these cells remain acetylated with a double methyl group as they become these hematopoietic stem cells. As these hematopoietic stem cells commit towards becoming a B cell, H3K4 becomes triple methylated. It already having the acetyl groups present and those histones loosely associated now allows these genes to be expressed. This opens up the chromatin and allows transcription factors and RNA, I'm out of room here, RNA polymerases to come in there and activate those genes. Now, I'm going to have to write the rest of it up here. Sorry for the messy organization. Once they become B cells, heterochromatin forms around these genes. So you can see this is a fairly efficient process where you have these genes here, VPRE-B1 and gamma 5, in the hematopoietic stem cells. They are in this poised position, ready to be turned on once proper signals come along and signal the hematopoietic stem cells to become B cells. They leave the poised state and enter the active transcription state and turn these genes on. When they turn these genes on, these transcription factors will then activate genes that are necessary to convert these stem cells into B cells. Once the B cell is formed, remember there's no going back, so we wrap these genes up in heterochromatin so they can't be expressed. Okay, that ends this podcast. In this podcast, we started with cells in that blastocyst stage, with these cells in the inner cell mass. These were, remember, pluripotent stem cells. So we focused on how do we maintain them in this pluripotent state, and then how can they change so that we can start to form the 3 layers of the gastrula, the ectoderm, mesoderm, and the endoderm. And then towards the end of the podcast, we talked about how we could take the ectoderm and create nerve cells, and how we could take the mesoderm and create the blood cells known as B cells. If you have any questions about any of the details of this podcast, please make sure you come see me. If not, I'll see you in class. Bye.